Hi everyone, welcome to the next video. This video will be covering ATP production as done by glycolysis, or this kind of second step within our energy use when we participate in physical activity, in particular sport, that again is anaerobic in nature. So our anaerobic system, just as a recap, is ATP-PC, which would have been covered in the previous video, and this one is glycolysis. Now in glycolysis, we're starting to get into an area that's has many more steps, so it's a little bit more complicated in one sense than ATP-PC, but ultimately at the end of the day, we're generating ATP, and it's done through this enzymatic kind of process that occurs over, in this case, 10 steps rather than one like the previous stage. So what is glycolysis? When does it happen? How do we use it? Let's take a look. So again, this, as you can see, is a little bit more complicated, and I'll explain what this is. You're not expected to memorize all these steps. I'll highlight a few specific things that you need to know, but we'll cover that as we go along. Okay, so here is your 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 steps of generating ATP. Now, one thing you might notice already is our initial step here starts with glucose. So previously, we had this high energy state bond in the ATP PC system that was just sitting there ready to release the high energy bond once the organic phosphate left the molecule to help regenerate or generate ATP from ADP. Here we actually start with a glucose molecule. So previously, when we're talking about metabolic substrates and carbohydrates, this is the really important metabolic substrate that our body uses, glucose. Okay, so again, within the cell, we have this glucose molecule that's floating around, and what we need to do is invest some energy in order for it to be utilized. So we actually need to use some ATP before we can start breaking it down. Now, just a little note here. So if we have our glycogen stored in the body, again, if you remember from the previous lecture, glycogen is this hodgepodge of glucose molecules that are hitched together. So if that gets freed up into glucose, what can happen then is it just enters a step later. So it doesn't use one of these ATP molecules to kickstart the metabolic process. Okay, we continue on. All these words are different molecules throughout the steps. And so what happens in between each of these blue lines is an enzyme will change the molecule in some way, shape, or form to give us the product at the next step. Okay. So again, once we get here to this fructose 6-phosphate level, we need another ATP put in to continue us on into these next steps. Okay. And once we get to this point, the molecule actually splits into two. So this original glucose molecule that's been changed 1, 2, 3, 4 times by now gets split into two molecules, and from there we can see the process continues on as we go into the second half. So I just recalled that I actually had set up this a little bit easier to see in a couple of slides. So again, it was a little challenging to zoom in on your screen there. I'll just reiterate in brief here. So glucose, glycogens are two starting points here. You can break down glycogen into glucose. So depending on where you're entering glycolysis or this anaerobic energy system, you're going to need 1 ATP potentially. By the time you get to stage 5, you'll need 2 ATP. So the things in the green boxes here you'll have to remember. Okay, so again, we get our molecule split there, and in the second half, as we continue on, you'll see the same thing is happening to both molecules. It's just happening in parallel. Okay, so we'll just track along one pathway here without going over both, but know that if something's happening to the one molecule, it'll be happening to the other too as we go along. So we get to this other process, and this little instance here you'll need to know, should have a green box around it, but we'll cover what NAD+ is a bit later in the lecture. And we continue on. And this is, again, you kind of need to know where we start getting energy from. So this step here, you start getting 2 ATP out of this, all right? So again, that's both sides added together. So here we have our 1 ADP being turned into ATP. Process continues on, and then here at step 10 again, enzyme comes in, helps capture some energy being released to make ATP from ADP. Okay. Now, the highlighted pyruvate molecule here at the end of the pathway is important to know, because pyruvate is this, I want to say byproduct, but it's a product of glycolysis. Now, a number of different things can happen here at this stage. Well, really, 2 things, but it kind of dictates what energy system is starting to be utilized and in what manner. Okay, so one of the things that can happen is this pyruvate molecule can continue on, and let's say again it's an anaerobic system, so in the absence of oxygen, no oxygen. So maybe the effort level dictated by this particular activity demands that there's not enough oxygen available to create this ATP. Therefore, what will happen is, again, this last step will occur and a lactate molecule will be produced, okay? So one of the things with lactate molecules is we'll commonly know that from the term like lactic acid. All right, so that's essentially where that term comes from, okay? Now, one thing I want to dispel and kind of put out there, the presence of lactate or lactic acid in the muscle or blood or whatever is not what makes you tired, is not what brings fatigue. It does not give you sore muscles per se, okay? Like, I just want to quash that right now. So everything you think you know about lactic acid is not exactly true. There's definitely a better explanation for it, okay? And the best explanation is previous research done for lactic acid and lactate generally revolves around correlating its presence to the real factors, and we'll cover this. We spoke about it a bit before, so hydrogen protons, inorganic phosphate. So those are now believed and supported to be the more pressing factors to cause fatigue in muscles or lack of force production. So not lactic acid, okay? Let's just trying to clear the air about that now, because that is a very common, still persistent thought within the health and fitness industry for sure. Okay, so we spoke about this one path ending up in lactate, so that's our anaerobic system. But the really, one of the key points that you want to remember is that pyruvate in certain circumstances will get converted into something that we'll cover when we go to the aerobic system, and then can be used in an aerobic energy production system. Now, this is really key, and I want to try to make it as clear as possible. Glycolysis is not an aerobic production of ATP, okay? It's anaerobic, no oxygen. But it does make the fuel for, or one of the sources of fuel for, our aerobic metabolism. Okay. So again, try to keep that in mind. So if we look at this whole process, we see this little net down at the bottom. So you get this net glucose and glucose from glycogen. So depending on which source we're using, at the end of the day, based off our input ATPs, so remember we had to put in some ATP, and based off the amount of ATP we get out here. So you can see that's there's 2 ATP here, 4 ATP. So if we just start with the glucose, we needed to put in 2, we got out 2, so we net. So 4 minus 2 is 2, so we get 2 ATP in the end. If it's coming from glycogen, so stored glucose in the cell, we actually get 3. Okay. So here we are, just what we just spoke about. It's a 10 enzymatic reaction. We get our 4 ATP technically, but we net 2 out of the process. 2 pyruvate, which may go on to lactate, may go on to our aerobic respiration or metabolism, for NADH out of the process. Okay, yeah, so it's an either or there. So glucose, where do we get that? How do we get that into the cells? Well, that comes from being freed up, again, from a number of different sources in our body, but can go into our bloodstream as simple sugars or blood glucose, or it'll be in the muscle already stored as glycogen. And this process of glycolysis, very similar to the ATP PC system. Just if you need a landmark in the cell to be thinking about where this is occurring, it happens in the muscle cell itself. Okay, great. So glycolysis, again, to reiterate, is anaerobic. That molecule that I was saying that pyruvate can turn into is called acetyl-CoA. It can enter the aerobic pathway. We'll cover that a bit later on. And again, if it's converted to lactate, well, it's kind of done. It's now not usable for aerobic systems. It actually has to go through this process called the Cori cycle, which involves the liver. It's called gluconeogenesis. So if you think of lactate as kind of like maybe a savings account in a bank, and you have to go to the bank and make a request. There's this whole other step and process where we need to make new glucose out of that lactate. Okay, and so that's what the Cori cycle is. It's a request to take this molecule, change it back to glucose, so we can have new glucose generated, genesis. Okay, gluconeogenesis. Now, that was just two of the byproducts of glycolysis that I spoke about already and that we covered. There's this other product that we've spoke about in the ATP PC system, but again is a cause of fatigue and lack of power, hydrogen. Okay, so again, these hydrogen protons changing the pH, bogging down our energetic systems, or in a manner of speaking. Okay, so we spoke about those ATPs, but the hydrogens we produce, they need to be removed. Okay, so there is a process to kind of be recycled, I guess you could think of it as, where all these hydrogens start slowing up our system. Well, our body realizes this and tries to figure out ways to get rid of it. Okay, so we do that through that molecule that I mentioned and pointed out a few times, the NAD, which then picks up a proton and shuttles it over to our aerobic metabolism systems. Okay, so again, I generally think of NAH or NAD as like a taxi cab or an Uber or something, right? So it'll pick up the hydrogen and shuttle it off to another system typically. Okay, if, for example, our aerobic system is too busy, too full, whatever, then the Uber can't drop it off there. Can't drop the hydrogens with our beta oxidative aerobic metabolic system. Well, then what ends up happening is it comes back to pyruvate, and then that's when we get lactate buildup. Okay, so if our aerobic system's too overrun, we start filling up lactate pretty quick. All right. And so, sorry, my last point to help illustrate and kind of solidify that. So NAD is the shuttles. So they're the taxi cabs running between glycolysis and aerobic respiration. And then if aerobic respiration is too full, say that's the hotel, you're traveling, you get there, it's too full, well, you got to go somewhere. So the pyruvate acts like a sponge and just holds on to it, kind of sucks it up as lactate, and then it's stuck there. Okay. The analogy kind of ran short there, but I hope you get the point. Okay, so just to highlight where this NAD is occurring, we got two stages, right? So here is where the shuttling occurs. So it'll grab the NAD and then shuttle it over to the aerobic system. Or if, again, we have our NADH now, so it's carrying the extra proton, the hydrogen, it'll unload it onto lactate. Okay, and that's how we deal with extra protons through glycolysis. Now, we obviously want to train this system. Yeah, sorry. So we want to train our glycolytic system, and one of the ways we can do that is the enzyme. So just as a little refresher, I won't go all the way back, but we if you recall here, every blue line, there's an enzyme at work. Okay, every single one of these, and it's just to slowly convert the shape, transfer certain energy maybe, or break it down into a molecule that then can later be leveraged for using energy production. Okay, so it's this multi-step process. There are specific enzymes that are trainable and that help in more, let's say, than others in this particular process. And again, when we're thinking about exercise physiology, we want to know what exactly is changing inside our bodies, physiologically speaking, that allow us to perform better. So when we put a stress or strain on our body, and then it tries to adapt by getting stronger to be able to overcome that stress later in the future, what training adaptations happen? That's what we're asking here. Okay, so 3 enzymes in particular you will for sure need to know. Glycogen phosphorylase. Now we just look at the wording for a second before we even actually know where and when it's happening. We already get a sense of what it might do. So glycogen, if you remember glycogen, that's the stored glucose in your body. Phosphorylase, well, phospho, that's perhaps to do with phosphate groups. And lase, if we recall from previous lectures with ases, well, those are enzymes that chop things off, right? They break things to lyse or to lase. So what does it do? Well, it breaks down glycogen into glucose in the cell. So this gives us our first step. We'll take a peek at it and come back. I incorrectly have the arrow pointing to glucose here. It's actually this step over here. Okay, so circle all of this glycogen into glucose. That's glycogen phosphorylase right there. Okay, right there. So that's where that step happens, right at the start. So you can imagine already if we train for, let's say, more of those enzymes or enzymes to be increased in their rate of capacity, right at that step, we're going to already net more ATP. Because remember, when we go from glycogen to glucose, it skips an input of ATP if we're going to go straight from, say, blood sugar to the first stages of glycolysis. All right, next enzyme is phosphofructokinase. It's a mouthful. People often say PFK. And the reason for that is right here. It's kind of when the first one of the first ATPs get brought in, and it is used at this step here. Now, why do we care about, that should not be glycogen. That should be phosphate, or phosphofructokinase, okay? So why do we care about phosphofructokinase, or PFK? Well, it's a rate-limiting step. And if you think back when we first were learning about enzymes, a rate-limiting step is a step in a multi-step sort of pathway that is the slowest. It is the slowest chain in the link. So that's why we care about that, because if we can increase the rate that a rate-limiting enzyme works, either by having more of it around or have its rate increased, what can happen is the whole process speeds up, right? You can think of like a team relay. If everyone has to finish the race at the same time, what dictates the pace? Is it the fastest runner or the slowest runner? The slowest runner will dictate the pace for the entire group, right? Same idea with PFK. And last but not least, lactate dehydrogenase. So lactate, dehydrogen, so getting rid of hydrogen, genase, that's another chopping sort of phrase. And we can look here at the last step when we have NAD giving up those hydrogens to our pyruvate to become lactate. Okay, so again, those 3 enzymes you'll definitely want to know. And how do we train glycolysis? Well, we're really looking at if ATP PC was to 30 seconds, glycolysis goes up to about 3 minutes. Okay, so any fairly intense activity you can maintain at high intensity for 3 minutes, you're really talking, it's, well, 2.5, I guess, to 3 minutes. So we're really talking about the glycolytic system, okay? So if you think about sprinting for that duration, some endurance does, but we'll also increase this depending on the intensity that you're training at. And resistance training or strength training will definitely be affected by the glycolytic system. Okay. Yeah, and when we're looking at results of adaptations for glycolysis, this is more like a research aspect or a critical thinking aspect if you're, say, a kinesiologist in the future or want to read studies about glycolysis, is that things like resistance training, interval training, etc., that improve the glycolytic system also tend to increase muscle size, so muscle hypertrophy. So the bigger the muscles get, usually produce more force. But a lot of the things that you would measure for glycolysis to see if that was improving also gets affected by... So a few other adaptations that'll happen with glycolysis include changes for intramuscular, or as it's noted here, IM, glycogen storage. Okay, so oftentimes having more glycogen present, as we saw earlier, anytime we have more glucose in the cell, that's a good thing. But the interesting thing is usually we get the best adaptation for intramuscular storage of glycogen through endurance sport. So activities that, let's say, take over 3 minutes at enough of an threshold level of physical intensity, right? Now, you can get improvements to glycolysis and glycogen storage, in the muscle through anaerobic activity. So, for example, like resistance training, sprint work, that sort of thing. But it heavily depends on the length of the training program as well as the type of training. So you really need, say, longer sprints of, let's say, repeated bouts of 2-minute running intervals versus, say, vertical jump repeats for 10 seconds. Okay, so that makes a big difference. And the idea behind the length of training program, as well as the longer that someone engages in these activities, repeated intervals, for example, you're going to have a greater duration, therefore a greater stress or training load on this individual, again, based over the time of the training program, versus something like endurance sport, where you get that time stress or exercise training on the individual, say, in one bout of 2 hours worth of exercise versus repeated intervals over time. Okay. Right. And then last but not least, as far as training adaptations with glycolysis, is we find adaptations in a buffer system. So if you haven't come across the idea of a buffer, the idea is buffers are kind of like this. If you're having a tug of war and you have two people on either side of you pulling a rope, you'd be kind of in the middle, trying to make sure no one individual really wins more than the other. Okay, so kind of like this elastic band or bungee cords or whatever, super bands tied to the middle of this rope. So the idea is, again, this system, the buffers, whatever the system's buffer, or whatever the buffer is buffering, whatever system it is, it will prevent any one extreme or the other. Now, eventually, buffers and the system that they're in can be overcome, but the goal of them is to maintain a little bit more of an adherence to equilibrium, or this middle ground between the two. Okay. So where do these buffer systems come into play, and how does it help glycolysis? Great question. I'm glad you asked. So with the glycolytic buffering system, one in particular is hydrogen ions. So we already spoke as to why that might be beneficial. Again, hydrogen ions, floating in the cell, really wreak havoc on the pH. The pH will wreak havoc on the enzymes that help us create energy for the activities we're doing, right? So, and that'll also come into play for recovery as well, post-exercise. Yeah, and there's a number of different systems here that happens with. So there's, let's say within the muscle, we have proteins, different phosphate groups, and something called bicarbonate, which is really important for blood. We'll talk about bicarbonate when we get to the cardiovascular system, for sure. And yeah, and so endurance activity for sure will help increase these buffer systems quite dramatically. But again, returning to this concept of how long are you doing your sprint work, say in intervals. And sorry, sprint work's just top of mind as an example, but you can have any activity with some sort of, let's say rowing for high intensity for 3 minutes, and then have repeated bouts over time. Or with endurance, again, a high enough training level or threshold for an hour, 2 hours, 3 hours, whatever the training session is. Okay, so both of those will help decrease muscle fatigue and decreases in power over time. Okay, so I just wanted to summarize here a little bit for you. Bicarbonate, that's the main one that I want you to remember. Phosphate, a little less important, and same with some of the proteins as buffering systems within the cell, but also just important to know and be aware of. But as I mentioned, we'll return to bicarbonate quite a bit. Okay, and so that's your lecture on glycolysis, your second anaerobic system. As far as producing ATP, we only have one that remains, and we'll discuss that next chapter. Cheers.